Okay, so here's a figure you've seen before. This was a figure that we discussed when we were talking about plant mineral nutrition. And we talked about the fact if we think about changing the concentration of mineral nutrients, say nitrogen in the soil, that there are regions where the concentration limits growth, regions where the concentration is optimal for growth, and regions where the concentration may be toxic for growth. Okay, and this is true for everything. So let's imagine a scenario. And this scenario is for everything that in any way interacts with plant growth, we adjust the concentration of it to be in this optimal range. Okay, so for nitrogen and temperature and pH and water availability and sunlight and everything. Okay? What determines how fast the plant grows under those optimal conditions? Yeah, so basically it's physiology, right? And it's going to be something related to diffusion or something related to the rates of specific enzymes or maybe the rate of DNA replication, something physiological, something internal to the organism, to the cells, is going to determine under optimal conditions how fast the plant can grow. So now let's imagine taking one of these external parameters, nitrogen availability, and putting it in the toxic zone or in the limiting zone. What's going to happen to the rate of plant growth? It's going to slow down, right? Something now new becomes limiting. This should be our functional definition of plant stress, right? Stress occurs when an external parameter imposes a new limitation on plant growth. And so what I want to spend most of the time thinking about today is thinking in general how plants respond to changes in external parameters. Changes in temperature, changes in light, changes in nutrient availability, changes in water, the sorts of things that we've been talking about all semester. But I want to spend most of the time doing it in a very general way. Not thinking about water specifically or light specifically, but let's think about the range of things that a plant would need to do. Okay, so let's just make a list of the various things that can contribute to plant stress, and let's do it in some organized way. So we'll start off with, let's say, atmospheric chemistry. What changes in atmospheric chemistry could impose limitations on how plants grow? Say that again? Yeah, okay, so CO2 limitation. What else? Well, sunlight's not really part of atmospheric stuff, right? It goes through the atmosphere, but let's not include it in the same category as CO2. Under what? Okay, water. And now we're talking about the amount of water in the atmosphere, not the amount of water in the soil. Okay? So that's humidity, right? What else? Oxygen. And let's stop for a second and think about CO2 and oxygen. Do the concentrations of oxygen and CO2 in the atmosphere change? Change enough to directly affect plant processes? No, basically no, except for increasing atmospheric CO2 associated with global climate change. But
what about the area around the plant, the changes that it produces around it? Is it going to decrease in CO2 during the day?
Yeah.
Physiology?
Yep, that's exactly what I want to get at. So it's not changes in the atmosphere that's really important. The changes that you're talking about are due to what?
Plant uptake.
Okay, plant uptake is taking up CO2, or if it's plant roots, they're taking up oxygen. But what does that got to do with it? Why does why does CO2 or oxygen become limited in those conditions? Rate of diffusion. That's exactly it. Right? So it's a physical limitation associated with, if we think about it as a simple model, you've got plant cells taking up oxygen or CO2, you've got a supply of oxygen or CO2 out of the atmosphere, and the medium through which those diffuse to get from the atmosphere to the cells is limiting the amount of diffusion. So in the case of CO2, it could be a boundary layer on the leaf where you deplete CO2 in that boundary layer, so it slows down the diffusion of CO2 through the stomates. In the case of roots, it could be waterlogged soil. Oxygen diffuses 6 orders of magnitude slower in water than it does in air spaces in the soil. Okay, so it's not the amount of CO2 or oxygen in the air that's changing. It's the coupling by the medium that connects the atmosphere to the plant. It's the diffusion through that medium that's slow. What else? More things we should add to this list. Say that again?
Well, atmospheric nitrogen, it's like legume or anything.
Yeah, okay, so in that particular case, although there's just so much nitrogen everywhere, that's not a limitation. We're missing some other important things. Well, temperature is not really a part of atmospheric chemistry either. It's sort of like sunlight. You know, it's in the air, but it's not really in the air.
Some pollutants.
Pollutants, yeah. Right, so give me some examples. Give me anybody. What are some pollutants, atmospheric pollutants that might affect plant growth? Sulfur dioxide. Ozone. Two more important ones. Nitrous oxide and nitric oxide.
Can you consider ozone a pollutant?
Say that again?
Is ozone a pollutant?
Absolutely. It's a reactive oxygen species that causes damage to plants.
Well, I know, but like, is it a pollutant? In the sense, like, you know.
Oh, okay.
Like,
So
it's bad, we know that, but like, a pollutant, I don't know.
How do we define pollutants?
I don't know.
Yeah, I mean, I think we sort of we link pollutants to anthropogenic sources. So, is there ozone that's produced naturally in the atmosphere? Yeah. Absolutely. Is there sulfur dioxide and nitrous oxide that's produced naturally in the atmosphere? Yes. Right. But it's the anthropogenic component of that. You know, that we've got 100 times more for sulfur dioxide in the atmosphere than there would be if we weren't burning fossil fuels. Right. So that's the component that we want to think about here. So all of these things, when the plant's exposed to them or when the plant doesn't get enough of them through diffusion limitations, can limit the growth of the plant. Okay? Let's take another category. Let's talk about soil chemistry. What are the sorts of things related to soil chemistry that could limit plant growth? Yeah, nitrogen, phosphorus. So let's put that into something more general. Nutrients. What else?
Soil structure, like we made.
Yeah.
Versus
Water.
Some water, yeah.
oxygen.
Oxygen, yep. pH, good one.
Cation exchange, but so pH.
Yeah, that's sort of pH and soil structure, soil composition, stuff like that, yep. So, we got nutrients on here. What about non-nutrient ions in the soil? What about aluminum and lead? Right? So, and those again can maybe naturally occurring or maybe anthropogenic. So, non-nutrient ions. One more important one. Think about plants growing along the side of Ithaca city streets in the springtime. What are they suffering from? Salinity, yeah. Salinity, which we would sort of say the total solute, ionic solute concentration in the soil.
Is it technically still a nutrient?
Say that again?
Technically it's still nutrients, right? If they have, like,
Is something a nutrient if the plant doesn't need it?
Would be non-nutrient ions?
Yeah, but these are talking about individual ones. Nitrogen, phosphorus. Salinity doesn't care which one it is. It's the total ionic composition, right? So if you make the ionic strength, if you make the solute potential of the soil too negative, what's that going to do to water uptake from the soil? It's going to make it a lot harder for the plant to take the water up, right? So the difference between salinity and these guys, these represent, so this could be like iron, or not iron, but lead or aluminum or something like that. The effects of individual ions. This is the sum of all the ions, the effects it has. Okay? And correct, the effect that this has is on water potential, water uptake in the plant. Right? So we can say that really this is affecting the availability of water in the soil. Okay.
Would competition from other plants be oh no, that's not competition.
Well, it would certainly affect the soil chemistry by decreasing the availability of some of these things, right?
Yeah.
Or
Okay, yeah.
Or by iron plants that produce like poisons to other plants. Or
Yep.
aloe.
Yep. Allelopathic and little chemical
Yeah.
compounds, yep. Yep. Okay, so what the thing that's holding this list together from some of the things that you just mentioned are these are all abiotic, right? Certainly biotic things are going to contribute as well. Okay, we've still got a few more things that we need to add on here, both of which have been said. Temperature, high or low, and light. Also high or low, and in some cases like quality. Right? So for example, under the forest canopy, most of the light that's usable by photosynthesis has been absorbed by the leaves above. Okay, one more we need to add this that we don't normally think about, but it turns out it's actually relatively important in plants, and that's mechanical stress. These are things that are like caused by wind,
People stepping on.
snow, yeah, the animals stepping on them, even hail. The animals stepping on, I think it would be biotic. So, although we're not going to talk about it, I'll mention it just briefly. There is a response to mechanical stress. Anybody know what it's called? Thigmomorphogenesis. Thigmo, touch-oriented, touch-related, yeah. So here's a simple experiment. Take a plant, rice is a good example, and grow rice plant in an environment where there's no air movement, nothing touches it, it's in a perfectly still environment. The rice plants grow about twice as tall as they normally do. Touch the plant once, and the growth rate slows down. So how many rice plants, or any plants, grow up without ever being mechanically perturbed by their environment? None, probably, right? It'd be very, very rare. Right? So this is an interesting one to think about from an evolutionary perspective. They're always being moved around. So why would it grow faster if it's not being moved around? It makes sense why it might grow slower if it does get moved around. Can you think of why?
You talked about plants falling over.
Yeah, that's basically it. Right? So tall, skinny plants tend to fall over pretty easily, or more easily. They'll be blown over by the wind more easily, or knocked over by rain or hail more easily. Right? So thigmomorphogenesis is something that every plant responds to. You can't think of a physiological condition where it wouldn't be exposed to that. But it does represent a response to mechanical stress.
Do you think that the slowing of the growth rate can also be related to competition if it senses Although that wouldn't take into account movements.
If the other plants were touching it, certainly. But if the other plants weren't touching it, but their roots were interacting, for example, or competing in the soil, I don't think there'd be any response there.
Yeah, on that note, I was thinking, how does the plant differentiate between like itself touching, like the blades of grass?
Yeah, in a sense, it doesn't matter. Anything that touches, or even just wind blowing by,
Yeah.
That's enough to slow down the growth rate.
Okay.
Okay, so any perturbation of the plant, whether it's caused by movement of the air or movement of an organism or touching another organism, all those things will cause these responses. So it's an interesting one. I mean, let's not spend any more time on it. But it's one of those, it's another example, I think, of non-physiological treatments in the laboratory that give us clues about what's controlling the way plants grow. It's sort of backwards from most of the ones we've been talking about. Okay, so every single parameter that we're talking about here presumably has an optimal range. Right? And when we go outside that optimal range, these are the things that cause plant stress. So you guys probably aren't old enough to know this, but when Bill Clinton was president, when they were having big budget problems, Clinton one day got up and announced that he was cutting the U.S. Department of Agriculture budget by a huge amount because he couldn't understand why anyone was interested in studying plant stress. And he was thinking of plant stress, like the plant sitting down with a shrink or something like that. He didn't really even understand what plant stress was. Fortunately, whoever was the head of the Department of Agriculture sat him down and told him that this actually was important if he wanted to be able to grow crops well. Okay, so let's think about now. We've got a whole list of things that can cause stress. Let's think about how plants respond to them. Again, in the most general way. And although the textbook only sort of does this, I want to break this discussion up into 3 categories. I want to talk about immediate responses. And to define, this is not mentioned in the book. These responses are mentioned in the book, but this terminology is not used. Let's use as our definition of immediate responses that do not require the plant or the cells to do any work. And these sorts of responses are familiar to you. You've heard about them. We've talked about them. We spent most of the first part of the semester talking about them. So, for example, if we change the temperature, what's an immediate response going to be in the cell that requires no work on the part of the cell? What's going to change in the physiology of the cell that the cell doesn't do anything to bring about this change? Only the change in temperature. Yeah, rates of reactions. Rates of reactions. Fluidity of membranes. Right? Those things change even though the cell hasn't done anything. If we think about osmotic conditions, if you change the osmotic conditions outside the cell, the osmotic conditions inside the cell will change in response to that without the cell doing anything. Okay, so immediate responses are those that are occurring almost independently of life. I mean, obviously you've got to have a membrane there and things like that, but the cell is not intentionally doing anything to change its environment. It's happening directly because of what it'll be perturbed. Okay, that part of it clear? Right. Now we really need to make a distinction between 2 things that are often mixed up. Acclimation and adaptation. And let's start with adaptation because it's actually the easier one. Adaptation, we need to think about this in the sense of evolution. This is long-term changes in the characteristic of an organism in response to natural selection by its environment. So this is really a response to natural selection. There are some plants that are evolutionarily adapted to growing in full sun. There are other plants that are evolutionarily adapted to growing at the bottom of the forest canopy in deep shade. There are plants that are adapted to growing in desert environments, and others that are adapted to growing in very wet environments. Okay, so these are adaptations that occur over thousands of generations of the plant in order to adapt the plant for its average conditions. So this is an adaptation to average conditions. Acclimation are changes that occur within a plant, within the lifetime of a plant, that allows it to adapt to changes in its environment. So this is within one plant's lifetime. The term that we use when we were talking about, for example, sun-shade adaptation is phenotypic plasticity. If we talked about the plantago, the weeds out in the egg quad. If you look at the plantago that's growing on the plant science side of the egg quad in the shade, those leaves are broad and flat. If you look at the same species of plantago that are growing on the Warren Hall side, the leaves are much more narrow and they're growing more upright. If you take the plant that's growing in the sun and move it to the shade, the new leaves that emerge are broad and flat. If you take the plants that are growing in the shade and move it over into the sun, the new leaves that emerge are narrow and upright. That represents a phenotypic plasticity in the plant that basically extends the range over which the plant is growing optimally. We've seen lots of examples of this, changing the composition of the membrane, right? So if a plant is growing in warmer weather and then it continues to grow in cool weather, it may change the lipid composition of its membrane to keep the membranes functional at the lower temperature, or vice versa, cool plants growing into warmer weather. So these represent work on the part of the plant to change its composition, but those changes represent something that allows the plant to extend the region of optimal growth. So, is phenotypic plasticity a part of adaptation? Let me read you a statement from the text. If you read the text that night, I would have hoped that when you read this, bells and whistles went off in your brain. The ability of a plant to survive and prosper in any given environment is thus related to a balance between genetic adaptation and phenotypic plasticity. Think about that for a minute. The balance between genetic adaptation and phenotypic plasticity. So, are these two separate things? In a sense, yes, because this is something that takes a long time to develop. Does this take a long time to develop? No, it can be expressed within one generation. But the ability to show that phenotypic plasticity is an evolutionary adaptation, right? Don't lose track of the fact that those two both represent evolutionary adaptations. This is an adaptation to average conditions. This is an adaptation that recognizes the plant will normally be growing in a range of conditions, and that if it didn't change its physiology over that range of conditions, the plant wouldn't be as competitive. So we saw an example of this. When were we talking? Oh, this is in the review session last night when we were talking about red, far red reversibility in phytochrome and the role that it plays in sunshade adaptations. Do shade plants show red, far red control of plant height? That is, do shade plants get taller if they're growing in more far red light and shorter if they're growing in more red light? No, because they're adapted to be growing in the shade. How about sun plants? If you take a sun plant and grow it in deep shade where it sees more far red light, what does it do? It grows taller. It wants to get to the sun. So keep in mind that these two, although we can treat them as being separate, they both represent evolutionary adaptations. But this is an adaptation to the overall average conditions. The plant always grows or almost always grows in deep shade, or it almost always grows out in the open sun. Where these represent plasticity within that average condition, the ability to change if the sun plant happens to be in the shade, have it grow faster so that it can get its leaves up into the sun. Okay? So does this distinction make sense to you? Okay, that's an important one when you're really trying to sort out what's going on in terms of adaptations. Okay, so when we think about all these different changes that can happen in the environment, and we want to think about how the plants can trigger this phenotypic plasticity, how it can change its physiology in order to be more successful when the environment changes. The first thing that should be coming to mind is what? It's a hard question. Not enough information to answer it. The answer I'm looking for is signal transduction pathways, right? Somehow the organism needs to sense the change in the environment and then initiate a sequence of events, the sorts of things that we've been talking about for the last 4 or 5 weeks. Right? So one of the things we should be thinking about is how are these changes? Remember the list we had there? You know, we had sulfur dioxide and water content in terms of humidity and pH and nutrients and light and temperature and mechanical things. The first thing we should be thinking about is how does the plant sense changes in this environment in a way that's going to specifically tell it, change your membrane composition because it's too cold, or make more photosynthetic machinery because the sunlight is too high. How are these things sensed? Obviously, it's a signal transduction pathway. So what do we know about temperature sensors in plants? Do plants sense temperature? Absolutely. Right? They sense temperature. We know that they do because they will change their, for example, in cold hardiness. Plants that are adapted to living in colder environments. If you grow flowers or vegetables in the house, you know, from seeds that you're going to put outside, the first thing you do is you put them outside for a little while on cooler nights to cold harden them, because if you just plant them in the ground, you get a cold night, they die. Right? So that represents a change in the plant's ability to tolerate low temperature. But how did the plant know that it was low temperature?
Unlike indirect sensing or more indirect sensing, we have like my membrane is not as fluid, and
Yeah, okay, so that's a good point. So could the plant sense that the membrane is less fluid? How would that, give me an idea of how that indirect sensing could work. I'm not saying you're right or wrong.
there should be something, there must be something that tells the plant that the membrane is maybe not as permeable or more permeable.
Okay, so it could be related to permeability. Are there processes in the membrane that require things to be able to move around, that if they couldn't move around or they moved around too much, might cause problems for the plant? Yeah, I mean, membrane fluidity is important in a lot of different processes. Think of PIN1 proteins, right, moving around to change oxygen transport. Right? So membrane fluidity is important. Right? So let me give you an example of one that we've talked about that gives the plant the ability to sense changes in all sorts of things. Let's think about how the plant senses light. So we've talked about phytochrome. We've talked about blue light receptors and the role that these play in developmental morphological changes in the plant. But most of the physiological responses to light intensity, sun-shade adaptation, for example, are not triggered by phytochrome. They're not triggered by blue light receptors. The tall, short in sun plants is triggered by phytochrome. But the thick versus thin leaves is not triggered by phytochrome. It's triggered indirectly by two different things. Let's think about the light reactions and the carbon reactions, or the dark reactions. Let's think about what couples those two together, because remember, this represents light in, right? The light reactions are using the input of light, and the carbon reactions represent using light energy. And we talked about conditions where the light energy in was too low, and that limits the amount of CO2 fixation that can go in. But we also talked about the conditions where light energy coming in is too high, where photosynthesis is saturated. So let's think about what couples these two and how what couples them might be used as a sensor for the balance between the light and dark reactions, and therefore whether the plant is seeing limiting light, sufficient light, or excess light. So what couples the light reactions and the dark reactions? Stella?
The production of NADPH.
Yeah, right. So we've got the ADP going to ATP here, and the opposite direction here, right?
Yeah.
And the same for
For
NAD, NADPH, right? Same sorts of things. So what could the plant, in the scenario where ATP, ADP is the thing that's coupling, what could the plant sense to detect the balance between light coming in and light that's being used? What could the plant sense?
The ratio.
Yeah, the ratio of ATP to ADP. Right. That would, so if ATP was high, what's the conditions? Excess light, right? If ATP is low, then it would be limiting light. The problem with using ATP is there's lots of things that use ATP in the cell. And in fact, the ATP-ADP ratio doesn't change very much from light to dark because there's just so many things controlling it. There's transients, but it doesn't change very much. The thing that is actually involved in sensing the balance here, there's actually two things. One is pH of the lumen. So remember, electron transport, the light reactions, are pumping protons into the lumen. Those protons are used to make the ATP that are used up by dark reactions. So under conditions where there's too much light, what will be the pH of the lumen? Yeah, it's going to be lower. It's going to be more acidic, right? And under conditions where light is limiting, the lumen is going to be less acidic, right? So the pH of the lumen is one of the things that senses the balance between the light and dark reactions. The pH of the lumen is the main thing that controls non-photochemical quenching, that process that gets rid of extra light energy in the antenna before that extra light energy can cause photooxidative damage. Okay? So that's something that's relatively limited directly to utilizing light energy. Another thing that represents the balance between the light and dark reactions is the oxidation reduction state of plastoquinone. Remember that plastoquinone is an electron carrier between photosystem 1 and photosystem 2. It's a mobile carrier that can diffuse in the membrane. If the plant is receiving limiting light, every electron that gets pushed into electron carriers goes to ultimately to NADP to make NADPH. And the electron carriers tend to be pretty oxidized. Under excess light conditions, you're not using up all the NADPH that's produced. It accumulates, and there's no place for all those electrons to go. And so the electron carriers get reduced. And this oxidation reduction state of plastoquinone plays a huge role in regulating gene expression associated with photosynthesis. So sun-shade adaptation is largely controlled by the redox state of the plastoquinone pool. So I'm mentioning these things for a couple of reasons. One, it represents the sort of indirect sensors that we're looking for. We could imagine a scenario where temperature might affect intermediates in a biochemical pathway or an electron transport. If one step is particularly temperature sensitive, then the intermediate before that step will go up or down depending upon the temperature. So these represent indirect ways in which environmental changes could be sensed. So the truth is that for most of the non-light related things, we don't really understand the details of the sensing mechanisms. We understand that there are sensing mechanisms and that there are physiological responses, but the details of the sensing mechanisms aren't entirely clear. Okay, but there's another reason that I talked about light and photosynthesis here, and if you remember back from our photosynthetic physiology lecture, the key thing is to remember that light is a common thread that links together many different types of abiotic plant stress. And so here's a figure that's not in your book. It's one that I made several years ago to show the relationships between the various components of plant reactions. So up here in the upper left, we've got the light reactions driving electron transport, making a pH gradient, making oxidants and reductants. These things in the stroma are driving the Calvin cycle. But the thing we need to keep in mind is that the Calvin cycle is intimately linked to many other processes in the cell and in the plant. So it's linked to water availability through the stomatal conductance, the opening and closing of the stomates and the availability of CO2. It's linked to nitrogen metabolism because carbon skeletons, you can't accumulate infinite amounts of starch in the plant. Eventually, the products of the carbohydrate products of photosynthesis build up to the point where they can inhibit things if you can't make proteins and nucleic acids and things like that. Transport of these compounds, the sugar compounds, to other parts of the plant, to the sinks of the plant, play a big role. So if the roots are being inhibited, for example, by the roots being flooded and not being able to do as much respiration, then that may affect the sink strength of the roots, and that feeds back to affect photosynthesis. So the thing that I want you to take away from this is that what happens up here with the light reactions of photosynthesis, remember that these things can trigger formation of reactive oxygen species if there's more light being absorbed. And reactive oxygen species trigger all sorts of stress responses in plants. So, for example, you can cold harden your plants. So if you want to, you're growing your plants from seeds inside, and you want to get them ready for potential cold nights without actually even taking your plants outside, it's very easy to do. Go down to the drugstore, buy a little bit of hydrogen peroxide, pour 0.01% hydrogen peroxide into the soil of the plants, and in 2 days your plants will be cold hardened. Completely non-physiological. The roots never see hydrogen peroxide, but hydrogen peroxide is produced in the plant as a reactive oxygen species when the plant is receiving more light energy than it can use. And cold conditions that slow down the Calvin cycle lead to the production of hydrogen peroxide. Okay, so what I'm trying to get you to see here is that there are many different types of abiotic stress that link back to photosynthesis. And the response to photosynthesis, normally for too much light, is production of reactive oxygen species. And reactive oxygen species will, in turn on a lot of the adaptations that we talked about, a lot of phenotypic responses that allows the plant to survive better under those stress conditions. Okay with that? Okay, so that's the general introduction to things. And let's at least spend a little time talking about a couple of stresses. I'm not going to go through all of them they talk about in the book, because in reality, with this introduction we've just had, and with the physiology that we've covered the rest of the semester, what happens in these stress responses should be pretty straightforward to you. You should be able to understand them. There's nothing new in there. We've heard about this all in various other circumstances. Now we're just going to bring it together here. So let's think about responses to limiting water. And let's recognize that limiting water most commonly occurs because the losses of water from the plant to the atmosphere exceed the ability of the plant to replace that water from the soil, right? So it could be low humidity of the air, or it could be low water availability in the soil, or some combination of the two of those, okay? Either one of those will. All right, so let's think about our immediate responses. What are the things that the cells don't have to do any work that represent a response to changes in water availability? What sorts of things happen when water availability goes down?
Decreased water potential.
Okay. Why?
Because water's leaving the cells.
Yeah, water's leaving the cells, or water's leaving the tissues, right? Okay, so what are the consequences of this?
Lower transpiration rate.
Yep. How is that lower transpiration accomplished? Is it accomplished in a mechanism that does not require any work on the part of the cells? Should we put this response in this immediate responses rather than in the acclimation responses? Why?
Because it's not something the plant is doing. It's just the tension theory that is basically based on the water potential of the air around the leaves and the water potential of the soil.
Okay, yeah, you're heading very much in the right direction. So let's just say the soil dries down. Is that going to affect the rate of transpiration even if the plant doesn't do anything else? Yes, right? Because it's going to mean the water potential of the soil is lower, and therefore it's harder for the plant to get the water from the soil. And that's everything else being the same, that'll decrease how much water the plant is using. So that's one component of it. What's another component of decreased transpiration as the plant water potential goes down?
Well, it's not really an adaptation, but could you have formation of kind of the water cavitation, like the bubbles?
Yeah, I don't know whether you want to call that an adaptation, but yes, cavitation, something that decreases, anything that decreases the conductivity of the xylem would contribute to that.
Close and stomates.
Close, okay, so the stomates close in response to low water potential without doing any work.
No.
I see some yes and some no. Who says yes? Why?
Well, because if the water potential falls low enough, then they're not going to have enough turbo pressure to
Yeah, right, so remember we talked about, a couple lectures ago, when we were talking about stomatal regulation, we talked about hydroactive and hydropassive responses. The hydropassive responses is if the water potential of the guard cells gets lower, along with all the rest of the cells in the leaf, will the stomates open or close? Close. That can decrease transpiration. So is there an immediate response in transpiration that's due to stomatal closure? No making ABA, no nothing. Right? It's just the lower water potential of the guard cells that's going to close the stomates. Right? So this is what I mean. These are the sorts of things that all the information that you need to sort this out, we've already talked about. You may not remember it, but you go back and look it up. But you should at least be familiar with it. Yes?
So wouldn't the fact that the water potential in the soil, just that fact that those are the stomates cause lower transpiration?
Yes. Yes, that will do it too. That all by itself, right? So there's multiple components to this lower transpiration, but all of them are ultimately associated with the lower water potential. You could say another consequence of this is increased solute content in the cells. The cells aren't making or importing more solutes, they're just losing water. Right? That's going to raise the solute content of the cells. Is that good in terms of dealing with water stress? Is raising the solute content of the cells good in terms of dealing with water stress? Yeah, of course. It makes it harder for the cells to lose more water. So that's another immediate response to this, is increased solute concentration. Okay? In the completely inactive, no work part of this. Okay, let's think about acclimations. What are acclimations of plants? That is, now they're doing some work. Now they're turning on some signal transduction pathway that changes gene expression, that changes morphology, that changes development.
Increase root growth?
Yeah. And what goes along with that? Yeah, decreased shoot growth. So basically, this is a carbon allocation problem. But in addition to these things, another thing that happens that we talked about is leaf senescence. Particularly the older leaves. What's the advantage of that? Why, under water stress conditions, get rid of your older leaves? Say that again?
It's also have carbon allocation?
No. No, not really.
Decrease of Listeria,
Why is that important?
so there's much less transpiration.
Yeah, right. This is just decreasing the leaf surface area. So there's less water loss in the plant. The plant's sacrificing its older leaves to limit its water loss.
Why older leaves?
Why older leaves?
Because they're less photosynthetically active.
Yep. Yep. And so then when those leaves senesce, do they just fall off? No, then the plant mobilizes the carbon and the nitrogen and everything it can from those leaves and saves it in the plant to do, when things get better, to produce new leaves from. Right? So it's a sacrifice. The plant can't do as much photosynthesis under those conditions, but clearly we've already seen stomatal regulation, right? Which wins in terms of opening and closing stomates. We know that sunlight, blue light, causes the stomates to open, and low water availability causes them to close. If we have lots of sun and low water, which one wins? Water, right? So it's the same with allocation of leaf area. It's sacrificing the ability to do photosynthesis to short-term save water. And what's the plant's thinking on this? Why is the plant doing that?
wants to, if it seeks to go through the whole period. The leaves are, for most plants, of no use, but helping to produce offspring for the next year.
Yeah, let me rephrase the question. Is the plant's expectation that water is going to be limiting forever? Yeah, that's a key part of this. That there's variability in the environment, and if water is limiting, it can sacrifice its older leaves and save the carbon and nitrogen that it can in the plant. And then when water becomes available again, make more leaves. Right? So from an evolutionary perspective, if the plant never saw enough water, what would be the result?
That the plants that can't save enough water will die if they want to produce.
Yeah, so what do you end up with then?
Natural selection.
Yeah, you end up with natural selection for what kind of plants?
Drought tolerance.
Yeah, you end up with succulents, right? Cactus and stuff like that. Things without much surface area for photosynthesis. Right? So if these conditions persisted forever, then you see a different type of adaptation. You see an adaptation that the plant never produces leaves. Just got some needles and some photosynthetic surface areas, and basically what's going on is it stores water, like a succulent. But in plants where there's the expectation, because of in natural selection, water becomes limiting and then later comes back, this is a very reasonable response to it. It's a response that you can imagine would be selected for in an evolutionary perspective. Okay, so how does this happen? So one of the things that people were concerned about is the decrease in shoot growth? Is the decrease in leaf expansion rate? I didn't put that on here, but leaf expansion rate goes down. Should put it on here because it's an important one. So for the leaves that are made on the leaf primordia, they grow more slowly. And one thing that you could say is, well, under stress conditions, the plant's doing less photosynthesis, and therefore it has less capacity to grow, right? But this information tells you that there's something else going on here, that leaf expansion is slowing down much more rapidly than the decrease in photosynthesis. So it's not carbon limitation that is causing this effect. Part of it is right here, that there's a change in the allocation of carbon between the shoots and the roots. What's controlling this? What sorts of things should we be looking for to think about allocation of carbon between shoots and roots? In what sense? Plant hormones, but in what sense? What are those hormones going to be controlling? Sink strength, right? That's the key thing. Because what determines where this carbon goes is sink activity. So what that means is, in the meristems, the growth of the meristems in the shoots must be going down. Something is slowing down the rate of cell division, the rate of leaf primordia formation in the shoots. So the shoots become a weaker sink. Something is turning on the activity of the meristems in the roots. So the roots become stronger sinks. Right? So it's not phloem transport, because phloem transport only responds to the sinks. So whatever the signals are that are moving around are altering metabolic activity in the cells that produce the shoots, slowing those down, and increasing them in the cells that produce the roots. How about lateral root growth? What happens under limiting water conditions to lateral root growth? So remember, lateral roots, if this is a root that's growing down, lateral roots, they're ones that branch off of the main ones. What happens to lateral root growth? Does it go up or down? I'm hearing up, and I'm hearing down. Why does it make sense that there's increased root growth at all under limiting water conditions? Find more water, right? So which is going to help the plant find more water? Lateral root growth or lengthening the roots? Lengthening the roots. Think about it. What happens when the soil becomes dry? Basically what happens is the water table, the region of the soil that's got water in it, gets lower and lower. Lateral root growth doesn't help the plant find new water. It's lengthening the roots. So lateral root growth is very much inhibited under limiting water conditions because it's not helping the plant find new water, which is almost always down. Right? So it's another case. Natural selection has selected for plants to be successful under drought conditions that can lengthen their roots, not that it can extend the network of their roots. Right? Because the water table's going down as water becomes limiting. Okay, another important component of this decreased leaf expansion. If we look at this graph should be familiar to you. We're plotting turgor pressure on the x-axis and leaf growth rate. We could think of that as leaf growth rate. We could think of it cell expansion rate. And we're plotting these as a function of turgor pressure for well-watered plants and for plants that are grown under water stress conditions. Somebody explain to me the difference in these two graphs. Not why they're different, just tell me what's different. What is this telling us about the differences in the leaves between plants that are grown with plenty of water and plants that are grown in limited water? Does this figure look familiar to you? Have you seen this before? Remember we saw this in the chapter on growth and development, or the chapter on cell walls. So what's this intercept on the x-axis? What does that represent?
The turgor pressure that's needed to start the cell expansion.
That's right. So that's the minimum turgor pressure to allow cell expansion to happen. The term that the textbook used was the yield threshold. Right? When the turgor pressure is below that limit, the cell wall will not yield, will not stretch in response to turgor pressure. What about the slopes of the line? What do those represent? That one you should be able to figure out. We've got turgor pressure on one axis and we've got growth on the other axis.
You need a lot more turgor pressure to produce the same growth in lower stress plants.
Yeah, so for a given change in the amount of leaf expansion, you need a bigger change in turgor pressure in the water stress plants than you do in the well-watered plants. Remember we called that wall extensibility? How much does the wall stretch in response to increased turgor pressure? This wall stretches more. You get a bigger expansion for a small amount of turgor pressure increase. This wall stretches less. What determines these characteristics? The change in the yield threshold and the change in the wall extensibility. What properties of the cell dictate those things? Construction of the cell wall. Yeah, basically the construction of the cell wall. So how tightly, how many cellulose microfibrils there are, how tightly they're held together, and this has something to do with the effect of acid, right? Because acidification of the walls is one of the things that stimulates cell expansion, but also the presence of proteins like expansins and things like this. So what this is telling us is that in plants that are water stressed, that are not getting as much water as they need, the cells are less able to grow due to differences in the cell wall characteristics. Why is this beneficial? If we think about and acclimation. Why is this beneficial for the cells of the leaves not to be able to grow as much under water stress conditions?
Less area to lose.
Yeah, so it makes less area to lose water from. Yep.
If it would expand at the same rate than the not water stressed one, then it would grow much more during the day.
It would use up more of the water. That's a good one. Here's an interesting question. I don't know if you know the answer to this or not. Plants are mostly water, right? So let's say the total amount of water in a plant is 1 unit. Okay? We're going to double the size of the plant. Plant grows and gets twice as big. So now it's got 2 units of water in it. To accomplish that growth from 1 unit to 2 units of water, how many units of water did it transpire? Anybody know? Have any idea?
1 to 100 or something.
It's about 1 to 100, yeah. So if it grew by 1 unit, it transpired close to 100 units of water. Okay? So in the context of the answer that the cells don't expand as much because it limits leaf area through which water can be lost, that's the much more important one than water used to produce new tissues. This is not here to limit the growth of new tissues. Water used to make new tissues. It's there to limit new tissues that could lose more water. Okay? So what does that mean? What are the responses in the leaf cells that are causing this? What could cause an increase in the yield threshold and a decrease in the wall extensibility?
Lowering the specificity?
Yeah, so less proton pumping, right? How about changing the composition of the cell wall? If there was more cellulose microfibrils or more hydrogen bonding that held it together. Yeah, either one of those. Okay. So that tells us right there that in response to water limitations, it's not just making fewer leaves, but those leaves that are made are producing cells that don't grow as fast because they have less stretchy cell walls. That's an advantage for the plant because it limits the addition of new leaf area through which the plant can lose water. Does that have consequences for the plant when water comes back? Right, so let's say we have 5 days of drought, right? And so, or 10 days of drought. And over that 10 days, the old leaves fall off. You have less leaf initiation. You have less leaf growth. So you've got a bunch of leaves that have started growing but haven't gotten very big. Now the water comes back. Does this have a consequence for the recovery of the plant after the stress? Is it going to speed up or slow down the recovery? It's going to slow it down. Almost every plant response that happens to stress conditions, even though the expectation is that eventually that stress will go away. Otherwise, we have some different sort of evolutionary adaptation. The response slows down the ability of the plant to recover from the stress once the stress is over. That'd be a nice exam question, right? Explain how that works for membrane permeability or membrane fluidity or rates of enzyme reactions or any of these sorts of things. How does the acclimation of the plant to the stress condition affect how the plant will behave once the stress condition goes away? In almost every case, it slows down the recovery. Plants that haven't been stressed will adapt faster, will change faster. Right? That tells you that the cost that the plant pays for slower recovery must be less than the benefit it gets for dealing with the stress. Okay. Another thing that happens in response to water stress is not just the immediate effect of losing water and therefore increasing the solute potential, but in general, plants that are stressed for any length of time produce compounds that we call compatible solutes. Solutes that can accumulate and lower the solute potential, make the solute potential more negative. More negative solute potential means it's harder for the cells to lose water, but compatible in that they don't cause other problems. So a very common one is the amino acid proline. Plants that see short-term water stress often accumulate proline, although there's lots of other examples here. Glycine betaine is another one that accumulates. So these compatible solutes don't interfere with all the normal things that the cell does, but lowers the solute potential, so it decreases the likelihood that water will be lost from cells. Interestingly, the most common compatible solute in life is not on this list. The most common compatible solute in life is a disaccharide trehalose. It's two glucose molecules linked together. The place that we see trehalose in plant kingdom is in a few groups of mosses called, they're club mosses. They're often referred to as resurrection ferns. So Selaginella, I wish the conservatory greenhouse was still there because there used to be some nice Selaginella plants growing in there. Selaginella, if you stop watering it, it dries down. It turns kind of a brownish color. And if you take these fronds and rub them between your fingers, they basically crumple into dust. You pour water back on, and within an hour, the plant's doing photosynthesis again. You can dry it down almost completely. In a sense, this is like a seed, right? At the end of the last stage of seed maturation is drying down, losing most of the water. We talked about compatible solutes being produced as the result of the late embryogenesis, abundant genes producing enzymes that make these compatible solutes. Trehalose, if you look in insects, in prokaryotes, in many animals, brine shrimp is a good example. Anybody have fish? Feed them brine shrimp? You buy brine shrimp frozen, dried. They're completely dried down. You dump them into the water, and in half an hour, they're swimming around just fine. Trehalose. Brine shrimp accumulate about 30% weight trehalose when you dry them down. Same with Selaginella. It accumulates a lot of trehalose. No. Do we have trehalose? Yes, we do have trehalose, although we're not really adapted to drying down. Right? We don't do too well when you dry us down. Right? But there are lots of insects, for example. There are some organisms some animals, frogs and fish that get trapped in ponds that dry up completely in the summertime. They produce a ton of trehalose. Interestingly, until recently, trehalose wasn't even thought to be present in plants. Any of you read the poster outside my office door? So I work with a group of scientists over in biotech who work on manipulating rice to overcome stress responses. And so when they first started talking about what genes should we put in rice to make it more stress tolerant, the first thing I said to them was trehalose. There's two enzymes. In fact, in some bacteria, those two enzymes are linked together into one enzyme to take glucose and make it into trehalose. So put these enzymes into rice, and rice becomes very stress tolerant. So I showed you, here's a picture right at the beginning here. Let's go back. There you go. So here is wild rice, and here is rice that has been transformed with enzymes that can make trehalose. Those plants were withheld water for 4 weeks and then given water back. So you can see the normal plants look pretty dead, and the ones with trehalose in it are growing just fine. They didn't grow much while they were water stressed, but they didn't die, so you give them water back. Okay, so this is a cool thing. Sounds really neat. Sounds like what trehalose is doing is being a compatible solute that's basically making the rice plants behave like Selaginella or like seeds. Wrong. In the transgenic plants, trehalose didn't accumulate at all. So this would be 25 or 30% trehalose by weight when it dries down. The rice plants were 0.01% trehalose. Turns out that trehalose is in every plant. All plants make trehalose. Every plant that we studied has the genes to make trehalose. But trehalose never accumulates even under water stress conditions in plants. Turns out that the role that trehalose plays, it's one of those metabolites whose level changes under stress conditions. So that changes in the amount of trehalose is one of the things that plants use to sense stress. Increases in trehalose levels turn on all sorts of stress responses. So whether it's chilling stress, heat stress, drought stress, nitrogen stress, trehalose, upping the amount of trehalose in the plant turns on all the plant's natural responses to those guys. So if you, next time you walk by my office, look at the poster outside the door there. It gives a nice explanation of what's going on here. So the reason I'm talking about this is because it tells us that plants have taken a different approach to using trehalose than all the rest of organisms in life have. It's not a compatible solute, but it is a signaling molecule whose, when the concentration of it changes due to stress-related phenomenon, it triggers, it's a signal that's detected by a receptor that triggers a signal transduction pathway that turns on all sorts of different stress responses.
Is that the case in Selaginella as well?
No, in Selaginella, it's basically just a compatible solute. Okay? So as far as we know, it hasn't been looked at extensively, but the role that trehalose plays in plants is not, it does not play that role in other organisms. It plays the role of the compatible solute. Right? So in other organisms, under drought stress, trehalose accumulates to high levels to protect the tissues under low water availability. In plants, it goes from 0.001% to 0.01%, and that increase is a signal to turn on all the normal things that would deal with the stress. Stella?
So trehalose accumulates in like those other organisms.
Yep.
And that maintains their cells' current water amount?
No, not necessarily. It protects it in the absence of water. So think about what happens in a seed. Right? So seed, the embryo gets all developed, you make the endosperm or whatever, you fill the cotyledons with storage stuff, and then the seed dries down. It loses 95, 90%, 98% of its water. It's in metabolic stasis, right? And then when you add water back, assuming that the dormancy's been released, it starts growing again, right? It's the same thing here. Right?
So the cells don't die when they use all that water?
The cells become metabolically inactive, but they're not dead. If they were dead, when you added the water back, the plant wouldn't be able to grow. Right? So we can't do that to you guys. We can't dry you down that much. Although maybe if we infuse you with lots of trehalose, we could. Trehalose is now being used to treat meat with, to be able to store meat for a long time. Whatever. Okay, let's just finish up the water, because there's a couple things we haven't talked about in terms of acclimations, and we've got to say at least something about adaptation. Other acclimations to water stress that we mentioned but didn't really talk very much about. We mentioned the fact that there's facultative C4 and CAM. That is, under conditions where water is available, plants are C3. Switch them to water-limiting conditions, and they switch over to C4 and CAM. Give them water back, and they'll switch back to C3 again. So this is a physiological plasticity. You change the conditions, they'll change reversibly.
What
So-
plants have them?
What plants have these sorts of things? The C4 plants, the one that I'm most familiar with is Flavaria. There's a whole bunch of plants that are C4. Think about it. Remember we said that C4 evolved, I can't remember what the number of times is now, 37 or something like times independently. And in that evolution, there's lots of intermediates between C3 and C4. Some of them are facultative. Okay, let me take one more minute. Because now let's take what we've learned about acclimation, physiological plasticity, and apply that to what happens under chronic conditions, where a plant grows in the desert its whole life, or a plant grows in the swamp its whole life and doesn't grow anywhere else. This should be easy, because the extremes of this acclimation, this physiological or phenotypic plasticity, represent the adaptations that plants have to living permanently in those environments. So, here's an easy one. Plants that live in really dry environments, they're constitutively can. They have more root growth and less shoot growth all the time, right? They have decreased rates of leaf expansion all the time, right? So another link that we talked about between acclimation and adaptation is that adaptation represents typically the extremes of the phenotypic plasticity. So the phenotype you'll see of a plant that grows in drought all the time, will be very similar to the phenotypes of plants that show physiological plasticity to drought. The phenotype of plants that grow in the presence of lots of water will be the phenotype of plants that are growing in the presence of water when they have this phenotypic plasticity. Okay, that makes sense? That makes it a lot easier to sort out what's going on here.